Okay. I think another 3 more minutes we shall wait and we can start our seminar. We have another 1 more minute to start our webinar. We shall wait for a few more participants to join. Hope this is okay with the presenters. Bismillahirrahmanirrahim. Alhamdulillah Rabbil Alameen. Wassalatu wassalamu ala Rasulil Anbiya'i wal Mursalin wa ala alihi wa sahbihi ajma'in. Assalamu alaikum to every one of you, the participants and the presenters. Thanks for joining our InnoSem webinar series 2 with the theme on the microbiome. We have 3 presenters today. Dr. Hafiz from Kuliah of Dentistry, Dr. Zaima from Kuliah of Science, Dr. Faizul from INHART, Institute of Halal Research Station, from Gombak campus, IIUM. Without further ado, we will start our meeting with Surah Al-Fatiha. Thanks everyone, and we are expecting few more participants to join as well. I have few things to say before we start our presentation, inshallah. Number one is each presenter will present their presentation for about 15 minutes, followed by a question answer session. Audience can ask questions in the chat box, and also request to unmute the microphone so that you can ask the question directly. And after each presentation, the question answer session will start. And meanwhile, the presenters are ongoing with the presentation. You can post your question in the form of text, no problem at all. And another thing is we have also the live streaming on Marine School YouTube channel. So those who cannot join this presentation in the Zoom application, you can follow back with the live streaming on IIM Marine School YouTube channel. You're most welcome. And this is a recorded presentation. So it will also be, it is automatically posted in the YouTube, and no one will miss any of the presentation by the eminent presenters. So as I mentioned, there are three presenters. We will start the first presentation with Dr. Hafiz from Kulia of Dentistry, followed by Dr. Zaima from Kulia of Science, and Dr. Faizul from InHeart. Without further ado, please, I hand over the floor to Dr. Hafiz, please.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh. And a very good afternoon to everyone. So inshallah, I'm going to share with you my prepared slide presentation. Okay, hopefully everyone can see my slide.
Yes, it's perfectly visible.
Okay, Bismillahirrahmanirrahim. Alhamdulillah, Allahumma salli ala Muhammad wa ala alihi wa sahbihi ajma'in. أما بعد فإن أصدق الحديث كلام الله وخير الهدي هدي محمد صلى الله عليه وسلم وشر الأمور محدثاتها وكل محدثة بدعة وكل بدعة ضلالة وكل ضلالة في النار. First and foremost, I would like to thanks to my respected brother Dr. Akbar for your invitation, and then to all my colleagues who are joining this program. So first and foremost, yes, I have been given 15 minutes to discuss on this topic, and then I think this topic is a very, very interesting, because now we know we are in the middle of COVID-19, and on top of it, some of the laboratories is still closed. Okay, and then many consumables are still not arrived to the destination. We have been purchasing many things, but until now, some of them are still on the way. Some of them are still not reached to the lab, and then even some of the labs are still closed, so we cannot run the experiment. Okay, without further much ado, the topic given is something to do with COVID-19, which is COVID-19 and an impact of microbiome. My name is Muhammad Hafiz Azmi. I'm from Kulia of Dentistry, as well as from the RMC, IIUM Kuantan campus. Okay, so this is the overview of my discussion. Number one, I'm going to discuss about the current situation of COVID-19, which I think will give you some rough idea how relevant it is for us to conduct study or to understand about microbiome. And the second one is to a little bit understand about the definition of microbiome in general terms. And then finally, we are going to discuss a little bit on how COVID-19 has been impacting on the study of microbiome as well as the microbiome itself. So this is the current situation of what I call COVID-19. This data just extracted this morning. I just browsed this morning at what I call the website of WHO, World Health Organization. So what we found out that for the data as of 3:59 p.m. 17th of August 2020, so the number of confirmed cases were reached to 21,549,706. So this is a very, very big number. And then it is not yet reduced. The number is still increasing. Inshallah, I found out that the number is still increasing due to many factors. And of that 21 million, we have 767 people death just because of COVID-19. Okay, so very big number. And then the question is that, does it going to impact the microbiome? So what is the microbiome? The thing that we have to ponder later on. Yeah. Okay, this is the global situation. So how about in the Southeast Asia? Okay, let us narrow down a little bit. Just now it's about the world number. Now how about the Southeast Asia? We have about 3 million. Okay, roughly about 3,103,18 confirmed cases of COVID-19. Okay, and of this 3 million, so we have 9,212 from Malaysia. This is the data as of yesterday. On the 17th of August 2020 at 12 o'clock in the afternoon. So look at us. Where are we? We are in, those who are in Pahang, so we have 370 cases. Those who are in what I call KL, so we have here 2,544. In Selangor, where is Gombak? IIM Gombak, maybe somewhere here in Selangor, isn't it? So we have 2,000 cases over there. So do you think it is not important? Yes, it is very important, and you have to study about the microbiome. How does this impact in many places, including in ourselves? Yeah. So new cases as of yesterday, we have addition of 12 new cases. And then the mortality here, we have 125 since registered until yesterday. So this data is obtained from Ministry of Health, Malaysia. Okay, now we already understand the what I call the current situation of COVID-19 in Malaysia. We know the number is increasing. We know that we have 4 million of individuals having infected by COVID-19. Then how about microbiome? What is microbiome? How can we relate this to the current situation of COVID-19? So in general term, previously we call it as microbiota, and during my school age, some of us call it as microflora. Now we have a little bit more, bombastic word. We call it as microbiome. So microbiome in general, and in a term of a layman, we can say that it refers to the entire habitat. If it is in the oral cavity, it will be oral microbiome. If it is in the ocean, it will be ocean microbiome. If it is in the forest, we will become forest microbiome. So what does it mean by microbiome? Microbiome is defined as the entire habitat that include the microorganisms. It consists of bacteria, archaea, lower archaea, and then we have also viruses, we have the protozoans, almost everything. Yeah. So all these microorganisms, when we compile it together according to the genetics, okay. So and then we call this as a microbiome, a constitution of microbiomes. Yeah, that comprise of all the microorganisms. Okay. So let's move on to the next slide. Okay. Normally, when I talk about microbiome, microbiota, when you deal with people, particularly in medicine or in dentistry or in health sciences, they will always think about microbiome in the gut, microbiome in the oral cavity, microbiome in the lung, but they forgot that they are also microbiome almost everywhere. Okay, so let me focus one by one. So do you know that microbiome, when we talk about microbiome, there is in the condition of us as healthy people. So we have a terminology where we call it as a balanced ecosystem in the oral cavity, a balanced ecosystem in the gut. So when you have a balanced ecosystem, there is no, what I call the potential of having disease is a little bit low. But somehow, if we change a little bit of this balanced microbiome, what will happen then, we call it as dysbiosis. Okay, dysbiosis means imbalanced microorganism constitution in any places, okay, in the niche or area. So for example, dysbiosis in the oral cavity, it leads to Alzheimer disease. So we have here Propharomonas gingivalis. When you change a little bit the number of Propharomonas, so there will be a potential of having Alzheimer. Again, if you look into periodontitis, periodontitis is caused by Propharomonas gingivalis, Treponema denticola, T. forsythia, and a few other bacteria. Okay, so this is actually caused by the imbalanced microorganisms. Okay, and even caries. All of us have Streptococcus mutans. We have Lactobacillus casei in the oral cavity. And on top of it, we drink Yakult. Okay, we drink Vitagen that contains this Lactobacillus casei. You know that Lactobacillus casei is one of the factor of dental caries, but still we consume it. It's not a problem. It is because of we want to make sure that in our gut, the microbiome is balanced. Okay, so that is the reason. And then again, the dysbiosis of microbiome can also lead to diabetes, rheumatoid arthritis, colorectal cancer, esophageal cancer. This is taken from the what I call the one of the articles which I'm sharing with you in my website. Okay, so this is from the oral cavity. Now let's go into the next one. It is not only on our body, yeah. It can also be indoor air microbiome. Those are working with what I call the air. You take the sample from the air. Okay, so we call that as indoor. If it is indoor, it'll be indoor air microbiome. If it is outdoor, it'll be outdoor air microbiome. Now you know that microbiome can be everywhere. Okay, so this is one of the study. This study is about the subway in urban society. Okay, and then the next one, forest microbiome. Yeah, microbiome can also be in the forest. And then it is, when I talk about microbiome in the forest, it's not just specific, it's not in general, there will be a specific location, like rock surface, there is a microbiome of the rock surface, invertebrates, atmosphere, foliage, bark surface, almost everywhere. So you can see that when you talk about microbiome, it's a very, very wide what we call area to be, studied. Yeah, we can explore it. Yeah. Okay, so this is a forest microbiome. This paper say that there is a diversity, complexity, and dynamics. We have on the rock surface, the microbiome will be different from the litter, from the soil, from the roots, and so on. Yeah. Now let's talk about, okay, that is in the forest. Okay, how about in the marine? Now, because this is a talk for marine, even in the marine as well, there is a microbiome for marine. Okay, so microbial dysbiosis, according to this paper, says that when we change the, due to the temperature, due to the increased pollution, due to the microplastic and so on, so this will increase the host stress or decrease the host defense. When you have the change in this microbiome, the host stress increase, the host defense, what do you call that? Host stress increase, host defense decrease, will lead to dysbiosis, meaning the imbalance of microbiomes. Okay, so you have some of the bacteria die, some of the bacteria is, supposed to be not pathogenic, become pathogenic for the, what I call, in this situation, those are supposed to be good now become bad because of the imbalance. Yeah, so when you have this, you have disease host. Okay, so this is basically what we can see, what we can study in further, particularly in the what do you call, the ocean microbiome. Okay, and the next one, this is one of the things that I would like to share with you guys. Okay, this is very important. Okay, as of today, I extract this data from Scopus. Okay, with the keyword COVID-19 and microbiome. I just want to show you that the study on microbiome and COVID-19 is a very, very, very low. Okay, because that this just started a few, what do you call it, not just started. We noticed that this happened in December 2019, and now it's already August. It's already past about 8 months. But until now, there is only, with this keyword COVID-19 and microbiome, there are only 22 documents, obtained from this from Scopus database. Okay, if you go to other databases, maybe there will be a more number. But only from Scopus, we only have 22 documents, and most of the documents are from what I call that review articles. Okay, it is only from the, most of them are review articles. I would say that a very little amount, maybe 1 or 2, is actually from the research articles that relates between COVID-19 and microbiome. Okay, now let's narrow down. How about ocean microbiome or marine microbiome? None. Okay, none. So no one has been studied how does COVID-19 can affect ocean microbiome, ocean microbiome or marine microbiome. None. Okay, so this is from database Scopus, which I do a little bit of search around 10 o'clock in the morning today. Okay, and then from the, what I call, the articles that I have been read, from the 22 articles, I found out that some of the published articles can be included in discussion. Okay, but yeah, most of them, not most of them, all of them are related to health. Okay, none discuss on ocean or other area, other environment. So, but from the newspaper, from other sources, we can see that there are lots of impact actually that happen from COVID-19 in relation to the microbiome. Number one, it impact on the research itself. As I said earlier during my introduction, so what happen nowadays, we have a very limitation to access for samples collection. For example, previously my student, we are trying to understand what is the microbiome in the, what I call, in individual with like Down syndrome, yeah, with autism and many others. But somehow due to, and then the samples, we plan to collect it. We have obtained the IRAC, everything. We plan to collect it from one of the school in the rural area of Pekan. Okay, and then unfortunately due to COVID, and then until now we cannot collect the data because this what you call, these children are from the school, so we cannot get the permission to enter the school. Okay, as everyone of us know that school is a very, very sensitive area to enter for the collection of data from the oral cavity. That is number one. Okay, limitation access to for sample collection. Number two, limitation of working laboratory. As you know that I don't know at Kuliah of Science and other Kuliah, but as for my Kuliah, we have a limitation. Some of us cannot enter the lab. The students, the FYP students particularly, they cannot join the lab. They cannot enter the lab. They cannot run the experiment. Okay, and then if those who can enter the lab, there is a limitation until 5 o'clock, for example. So this is again being a very, very big limitation for us to conduct research and to understand, or to even to understand how COVID-19 can actually impact on the microbiome itself. Not only on microbiome, on several others experiments also been affected. Okay, the next one is that it also impact on the environment. Okay, if you can see during MCO, okay, it's very difficult for us to go out except to buy some groceries. During that time, it's a very clean. When you go to the when you go to a beach anywhere, there's a very almost difficult to find rubbish. But after MCO, now RMCO, PKPP, you can see that there are lots of masks everywhere. And then again, this mask, when you introduce something in the environment, you are introducing something that can attach by the microorganism. Then we have the microbiome of the mask. We are actually changing the microbiome in the environment at the beach or on the floor, everywhere. Yeah? So this is another potential thing to be studied. Okay, this has been affected a lot of us actually. And then maybe I hope that any of us from Kuliah of Science or even Kuliah of Dentistry or any other Kuliah, we can try to find out, we can figure out how are we going to solve these problems, particularly with the mask, which is thrown everywhere. I don't know. Okay, we have to think about this. Okay, the next one is, this is very common, it definitely will impact on the health. Like for example, with the PKP, less physical activities, all of us are staying at home, just order from somewhere, and then you get the food. And then when you have this, you have, your diet is changed, and then you are becoming stressed because of you cannot enter the lab and so on. So this change of the affect your what we call microbiome. Yeah, microbiome in the oral cavity, microbiome of your gut, and everything because of like what we call different change in the physical activities and diet and so on. Okay, so let me show you a little bit of the data. Okay, of the articles that I found out is very, very interesting. Number one, okay. So what people have shown that, okay, Al-Alfi and Al-Haddadi. So these articles, he showed that, okay, gut microbiota can modulate, okay, the complication in COVID-19. Because of what? They said that when, what they call COVID-19, they give some drugs. So what happened, okay, when they take the, okay, for example, here you see that zinc and chloroquine. So once people take the drug, what happened is that it changed the diet, it changed the microbiome in the gut, okay? That is why when people, what we call as opportunistic microbes, at the beginning, it doesn't cause any harm. For example, Candida albicans. If you have heard about Candida albicans, all of us have Candida albicans. About 75% of healthy individuals have Candida albicans. But when you consume fluconazole, okay, any antibiotics for so long, okay, and then, sometimes you stop it, what happened? It changed the microbiome in the oral cavity. Okay, it changed, it kills some microbes, but it allows some microbes to grow. So this is the same in the gut. Okay, so what happened then? This paper said that it may be because of the, it is not because of chloroquine does directly, treat COVID-19. It's because of you change the microbiome in the gut, which lead to the survival of an individual with COVID-19. That is one what I call hypothesis. They say that. And the second paper also saying that there's a potential of COVID-19 and there might be a relationship between COVID-19 and gut microbiome. What happened, the history is that COVID-19 is due to something that that is obtained from food. Okay, so they say that and then ingested or maybe inhaled and so on. But in this paper, okay, so this is one of the quite interesting paper. So it says that there is what I call a relationship, the potential relationship between COVID-19 and gut microbiome. Again, maybe we can study about this in more detail. So I have a very limited time. I have another few more minutes. Let me share with you. It is not only about gut. It is also about lung microbiome. They say that how people can get, how people become so severe. Some people are severe, some people are not. Some people are okay. One of the things is said that lung microbiome, when we change the lung microbiome, what happens is that it will trigger or it will affect the immune system of what I call in the lung environment. Okay, when you change the microbiome, it changes the immunity of a certain individual. Okay, high microbial density, low microbial diversity. So your healthy lung is supposed to have low microbial density, high microbial diversity. When you have the changes, Okay, this microbiome supposedly to prevent from infection, inflammation, immunity development, activates immune system. But when you make the diversity low, what happens is that you change the microbiome, eventually cause a dysbiosis. When you have dysbiosis and it increase inflammation, expression of certain interleukins like interleukin 6, interleukin 8, interleukin 10, which are responsible for some chronic infection, difficulties of breathing. So this is another paper. So let's go to the next one. Okay, those colleagues from Faculty of Dentistry. So here is another thing which is very interesting. This is about I just get this paper from British Dental Journal, one of the Nature under the Nature. So what we can say that this paper, this is like a general idea that's just been thrown out by one of the prolific microbiome researchers, where they say that there is a potential link between oral hygiene and severity of SARS-CoV-2. So the reason the reason is that they say that when someone taking care of their oral cavity, so what happens is that there will be they say that the pathogenesis of these microbes to the respiratory infection can be aspiration of oral pathogens into the lungs, and the periodontal disease associated enzyme may modify the mucosal surface to allow for addition of colonization of respiratory pathogens and so on. Okay, so what happened, I'm saying here is that there could be possibilities of the microbes that is actually from the oral cavity get into the lung. Okay, as you know that some of the microbes, say for example Staphylococcus aureus, it is from the oral cavity but end up in the endocarditis. Okay, so in infection dekat jantung kan. Okay, so this is another interesting paper, and then the interesting research that can be explored. Okay, last but not least, okay, this is for everyone, those who are listening to my discussion. Okay, so the topic is that about the most important challenges ahead of microbiome pattern in the post era of the COVID-19 pandemic. So one of the things that is quite interesting in this paper is that now we are using disinfectant. Where is it? Where is the? Okay, cleaning products during COVID-19 pandemic. So we are using disinfectant, recommendation to consume probiotics. Okay, so what this paper is saying is that, okay, when we use household cleaning products and, so this can also affect the what we call microbiome. We can imagine the microbiome on the oral cavity, microbiome on the skin itself. So you are changing this dramatically. So there are lots of things ahead that we can study. So yeah, I think that is enough for me. 15 minutes of presentation. So you can get this slide. I have provided this slide in my website. You just Google hafizazmi.com. The slide is there. And then the paper also there. You can just download it. I give you the link for the for my presentation, as well as the articles related, which I put it together in the in the slide. With that, thank you very much. I hope you get some idea of what I have been discussing. Thank you very much.
Thank you, Dr. Hafiz, and it is one of the interesting presentation, and you open up a lot of doors for research discussions and etc. You have one question asked by one of the member, Ahmed Faisal. Can microbiome become a possible predictor in future?
Okay. What do you mean by predictor? If you say that predictor means you want to, for detection of for detection of disease, or Ahmad Faisal, if I'm not mistaken, one of our colleagues from dentistry. Okay, so we can say that in certain disease, okay, we have different composition of microbes. Okay, so we have certain if you have oral cancer, we have certain composition. If it is in caries, there will be a different composition of microbes. For example, in caries, we have abundance of Streptococcus mutans. Okay, but in normal condition, the Streptococcus mutans will be very, very low. So in other words, we can say that there is a potential of microbiome to become one of the possible detection kit, for certain disease, which I think if you can predict, maybe you can predict whether these people will have caries in the future or not. Maybe you can predict as well if this patient if you have this certain microbiome, what will happen in the future if you change a little bit. That you can predict even for the marine microbiome, what will happen in the near future by changing in the microbiome itself. Maybe you can predict something in the future. So I think, there is a potential. There is a potential that microbiome can become a possible predictor in the future. That is my answer.
Thank you. If there is any question, maybe audience can pose. If there is no question, then I will move on to the second presenter, Dr. Zaima.
Okay, thank you.
I request Dr. Zaima to take floor. Dr. Hafiz, maybe you need to cancel your screen.
So can you all see the slide?
Yes, doctor.
Okay. So can I start now?
Yeah, please, doctor.
Okay. Assalamualaikum warahmatullahi wabarakatuh, and a very good day to all present in this webinar. First of all, I would like to say thank you very much to InnoSam for inviting me for today's webinar program. So I'll be presenting something totally different, okay, from Dr. Hafiz. Okay, I cannot top that with the COVID-19 thing, but I think it's as important as well for this topic. Okay, so I'll be presenting about bioprospecting actinomycetes from mangrove forests as new resource of antibiotics. So I am Dr. Zaima from Department of Biotechnology, Kuliah of Science. Okay, I think every one of you have heard or have been to mangrove before. And so mangrove actually constitute a large portion of the coastline in the tropical and subtropical regions of Earth, which is around 60 to 70%. And mangrove is known because they provide spawning and breeding grounds, nurseries, and habitats for many aquatic, semi-aquatic, and terrestrial animals. And the root system of mangrove trees also provide natural barriers that reduce impacts of storms and protect coasts from erosion. So in Malaysia, so the mangroves is actually less than 2% of the total land area. So it's around 641,896 hectares of mangrove forest in Malaysia, in which majority, which is the 57% in Sabah, 26% in Sarawak, and the rest 70% in Peninsular Malaysia. And Matang Mangrove Forest Reserve, which is in Perak, west coast of Peninsular Malaysia, is regarded as the best managed mangrove forest in the world because of their practice in conservation and charcoal production. So mangrove as habitats, right, for animals and plants, but they are also hotspots for microbial diversity. So microbial community plays essential roles in the functioning and maintenance of the ecosystem. Okay, so next is about antibiotics. So I think everyone knows what are antibiotics. So when you are sick, you're probably your doctor probably prescribe you antibiotics, okay, to cure a certain infection. So antibiotics are chemical substances. So they inhibit the growth of microorganisms. But then there's the issue here, which is the antibiotic resistant bacteria caused by antibiotic abuse. So it's basically a threat to human health because it counteracts the beneficial effects of the bioactive compound targeting the pathogenic microorganisms. So it will often resulting in treatment failure and severe health consequences. One example is like this MRSA, methicillin-resistant Staphylococcus aureus. Okay, it's a common infection in hospital. Okay, so because they are resistant to methicillin, so you have to treat them with a much stronger antibiotic. So another example of antibiotic-resistant bacteria is the vancomycin-resistant enterococcus, known as the VRE. So one way to overcome this problem is actually to come up with a new bioactive compound, okay, so that you can reverse and counter the spread of antibiotic-resistant pathogens. So this is where this actinomycetes comes in handy. Okay, because actinomycetes actually a large and diverse group of gram-positive bacteria with a high GNC ratio, and they are common soil inhabitants. So they are actually known for their ability to produce bioactive compounds. You can have antibiotics, anti-tumor, immunosuppressive, and anti-inflammatory agents, just to name a few of their activity. But in the environment, actinomycete plays an essential role in nutrient cycling and mineralization of organic matters. One of the genus, okay, the genus Streptomyces alone. Okay, if you look down there, you can see the colony morphology as well as the scanning electron microscope. This is how they look like on the plate and also under the electron microscope. So this genus alone accounted for almost 70% of commercially available antibiotics in the current market. So this just to show you how very important of this actinomycete is. So they can be categorized into two major groups. You have the dominant group, you have the rare actinomycetes group. So if it's in a natural habitat, okay, the Streptomyces and Micromonospora are among the dominant genus of actinomycetes. So when you do isolation, these two are the common bacteria you will get when you encounter when you did actinomycete isolation. But the rare one, okay, it has lower isolation rates and harder to cultivate due to their extremely slow growth. In our experience, okay, we have encountered one of the rare actinomycete which take about 20 days just to grow. Okay, so you really have to be patient and pray to Allah that you will get this rare actinomycetes. Okay, so why mangrove actinomycete? So why the buzz of this actinomycetes? One of the reason is that because of its soil bacteria, and because of their ability to produce bioactive compounds. So many research has been done, extensive research has been done on the terrestrial counterpart. And because of this, it led to the exhaustive actinomycetes strains and rediscovering of similar compounds. This is something you don't want to get. You don't want to get the same compound over and over again. So one of the solution is to actually to look for another location other than the terrestrial counterpart. You might go to unexplored location or underexplored location, like the marine environment. You might want to get your sediment from maybe 100 meters under the sea, or you want to go to extreme environment. So these are considered unexplored and underexplored location. So one of the underexplored is the mangrove forest. So researcher has considered this as a new resource of actinomycetes, which may lead to the production of novel bioactive compounds. So several novel genera has been described. Okay, you have this new genus being described from all over the world, from mangrove all over the world. For example, like Illuminobacter, Lysinium microbiome, Mumia, and Monacea, and numerous novel species has been reported. So this is actually a table highlighting the compound being produced by mangrove actinomycetes. You have the azalomycin, for example, okay, which is produced by Streptomyces, and it has an antifungal and anti-tumor activity. You have the bitrimycin, produced by Micromonospora, and it has antimicrobial activity. But if you look closely on the table, okay, you can see that the majority of the compounds are actually produced by these two bacteria, which is the Streptomyces and Micromonospora. But there are also other compounds being produced by the rare ones. For example, like this one, the nocardiatones A, B, and C, produced by rare actinomycete, Nocardiopsis, and they have antimicrobial and anti-tumor. Okay, and Selenisporamide A, produced by Selenispora tropica, and it has an anti-tumor activity. Just to show you that there are potential of new compound being produced by rare actinomycetes. So next, let us look at the mangrove actinomycetes. Research has been done in Malaysia. So I'm just showing you several locations. Pahang is quite extensive, basically. Okay, quite extensive research has been done on mangrove actinomycetes in Pahang mangrove forest with the discovery of novel genera like the Mumia flava, Monacea flava, and the description of novel species. So quite a number like Microbacterium, Mangrove, Sinomonas, Streptomyces, Mangroveosoli, and so on. So several research done from Pahang mangrove forest showed high diversity of actinomycetes. Some showed antimicrobial capabilities. And in Sarawak, they conducted research on diversity of mangrove Streptomyces with the description of a new species, Streptomyces colosenens. And in Matang Mangrove Forest Reserve, they did genome analysis of Mangrovemonas-like strains. But this is just a few locations, but I believe there are other locations, other mangrove forest location in Malaysia, maybe in Terengganu and Kelantan. It's just that not yet being reported. So I would like to share to you with, some of our findings from our bioprospecting program in Kuantan mangrove forest. So we did sampling 7 locations. Okay, we have K1 until K7, and we have K1, K2, and K3 located at the Kuantan mangrove forest. K4, K5, K6, and K7 located near to the sea. Okay, and we actually got a lot of actinomycete isolates, but we only focus on those with a very interesting morphology and bioactivity. So we found that the mangrove-derived actinomycetes isolated in the present location demonstrated considerable level of diversity. So we focus on the 52 isolates, okay, and distributed to 13 genera. We have the Deutzia, Micrococcus, Nocardiopsis, Actinophytocola, and of course we have the Micromonospora and Streptomyces being the dominant one. So the rest are considered as the rare actinomycetes. And we also noticed several of the isolates actually show antimicrobial activity against E. coli, Serratia marcescens, Staph aureus, Bacillus subtilis, and Candida albicans. Right, this one is actually just to highlight some of the potential isolates from our finding. Okay, and we also did detect the gene, biosynthetic gene, the NRPS and PKS1 gene. So these 2 genes is actually usually related to the secondary metabolite production. So it's very interesting if you have isolate that have this gene. So if you can see from the table, you can see the Streptomyces K101. This one actually has a broad antimicrobial activity and considered one of the potential to be exploited. So beside the Streptomyces and Micromonospora, we also have the rare actinomycetes. We have the Pseudonocardia, and we have the Nocardiopsis and Actinophytocola. Okay, these actually showed antimicrobial activities as well as and the presence of these 2 genes. And we are very excited to see this Actinophytocola because no research has been done. Okay, no report on the antimicrobial activity, no report on the biosynthetic gene. So this is considered as a potential actinomycete to be utilized for natural product discovery. So these are some of the images of what we got from our bioprospecting program. Okay, we have the Saccharopolyspora, we have the Gordonia, we have the Verrucospora, just to show you what does it look like from one each of it. And personally, this Actinophytocola takes about 20 days just to grow. Okay. So as a continuation of our project, we have TEM, isolation and purification of bioactive compounds from rare actinomycetes. And another colleague of mine is actually doing biotransformation of compounds using mangrove streptomyces. So what are the future direction? Okay, we were thinking what can we get from our mangrove forest, right? So mangrove forests basically provide opportunity for microbial natural products exploration. So we need to harness the mangrove actinomycetes fullest potential, perhaps not only for antibiotic production, but also maybe to use them in biotransformation, bioremediation process, for enzyme production. So the choice is endless. And the next would be the in-depth studies of micro-inhabitants of mangrove forests, so that we can have these metagenomic studies to actually understand and to have the overview, okay, overview of the representative microbes present in mangrove. And metabolomic studies, we talk about metabolomic studies. This is basically studying the secondary metabolites. We want to see the metabolic fingerprinting of the actinomycetes. And the current trend will be the whole genome sequencing and genome mining of mangrove rare actinomycetes. Because when you have the whole genome sequencing, you can actually identify the secondary metabolite gene clusters, which is very interesting to study on, and also the genome mining, so that we can tailor our natural product process, okay, through this by identifying the secondary metabolite gene clusters. Okay, so we have a lot of culture actually. So if anyone wants to collaborate with our team, so please do so. All right, so thank you for listening, and thank you again.
Thank you, Dr. Zaima. I open up the floor for the question. If anyone have a question, you can ask in the live chat, YouTube, as well as in the Zoom platform. Or if you still have the question in mind, keep it with you. Still, we have time, so that after the third presenter finish his presentation, then we can open up the floor again for the question, where you can ask the question to all the three presenters. I do have two questions, and I will ask Dr. Zaima.
Okay, no problem.
Not now. I will
later.
Audience first. Yeah, there is one question from Khairani Ikhwan. If you want to ask the question, voice also okay, but I will read the question, by the way.
Okay.
Other method that we can use to discover novel antibiotics other than direct isolation from environment?
Okay, all right. When you talk about direct isolation, what do you mean by direct isolation? Is it direct isolation of the bacteria, of actinomycetes or bacteria, or direct isolation of antibiotics?
Khairani. I will check it out, the Khairani name, and then open the, yeah, I will.
Okay, what can I say is that beside the traditional isolation of and purification method of natural product, you can actually use the bioinformatic tools to predict the what compound you can get, because in a normal behavior, in a culture condition, sometimes only one, but if when you do the genome sequencing, you can actually see they have a lot of compound that can be produced. It's just that a matter of optimizing the method to harvest the natural compound.
See, I think the answer is very clear to Khairani. Okay, let me ask Yeah, there is a response
Okay.
from the audience. Okay, I have a question, Doctor. Number one,
Yeah.
very simple, straightforward question.
Uh-huh.
Why the actinomycetes are more colorful? Number one. Number two,
Uh-huh.
uh, why the two strains are more common compared to the other actinomycetes species? Either it is isolated from the mangrove environment or from the other environment as well.
Okay. All right. The first question, why is it colorful?
Yeah.
Yeah, because they can produce pigment, but it will depend on what carbon sources you are feeding them. Okay, sometimes if you feed them starch, okay, it will become white. But sometimes if you feed them glucose, they will change the color. So carbon sources also have influence, as well as other components of the medium will also have influence in the pigmentation of the colonies. Okay, and this also the components is actually the composition of medium has also influenced the secretion of the compounds. So if you change, okay, if you change the carbon sources, the simple one, okay, you might get something, you might get compound A. But if you change, you will not get the compound A anymore. Okay, so it will produce something else. So you can play around with that. The second question is, why the two, Streptomyces and Micromonospora? So in terms of isolation, it doesn't matter what location, these two will dominate. Whether you isolate from the soil, whether you isolate from water, or whether you isolate from sponge, these two, the Streptomyces and Micromonospora, will always dominate the population. So that's why, because this is because they are more, it takes shorter time to grow compared to the rest. And because they dominate in the habitat also. So that's why it's easier to get them. So is this okay?
Yeah, it's very clear. Thank you very
All
much.
right.
That's great. If there is any other question, we shall wait for it. Okay, I think the audience can hold on to the question, and we will go with the third presenter, followed by the question answer session. Dr. Faisal from Gombak in Heart. Dr. Zainal.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Okay, I will share my screen now.
Is it fine, Dr. Faisal?
Can you see that my screen? Okay, wait. Because I'm using Mac, there's some something that I need to change. Okay, sorry. Okay, Dr. Akbar.
Is there any issue?
Okay, can I change the? I think I need to change my computer for a while. Okay, can you continue with that things?
Yeah, sure.
Okay, all right.
If there is any question to the two presenters, please post. I have one question to Dr. Hafiz, if there is.
Yeah. Yes, please.
Yes, fantastic. Okay, during your search in the Scopus directory, the word that was used is the microbiome as well as the COVID-19. Is there any other keywords that you use to derive the papers out of it?
No, so far I just used that too. So to make it relevant to the topic. But I think if I put it as microflora, microbiota, maybe the number will be increased.
Yeah, true. There's some when we add more keywords, then the search engine will work better. And also the SARS-CoV-2 is another keyword that we can use it as well.
Yes.
And but the issue that you raised up is very informative, really. like a lot of things to explore in terms of microbiome diversity in relation to the COVID-19 patients as well as the environment.
Yeah.
That's really informative.
Mhm.
We shall wait for Dr. Faisal and let's see. This is the issue when we use Mac. Dr. Zaima, I hope you cancel your screen sharing already, right? I think so. Fine. Dr. Faisal joined already. We should wait for his. Dr. Faisal? We shall wait for another few more minutes. Dr. Faisal is entering the Zoom meeting.
Assalamualaikum warahmatullahi wabarakatuh.
Perfect.
Okay, I'll share now. Okay. Assalamualaikum again. Sorry, there's some technical with my Mac. Okay, so I need to change and there's no more Antarctic background at my back. And now I'm back to Malaysia. Okay, so thank you, Inosam, for this webinar series, and also Dr. Akbar for inviting me. As the topic is microbiome, and it's already discussed by Dr. Hafiz and Dr. Zaima, different topics. Now is maybe we go a bit far, yeah, the polar regions, okay, with my title, Polar Microbiomes, Opportunities and Challenges. My name is Dr. Nur Faizul Hadri from International Institute for Halal Research and Training, IUM in Gombak. All right. So maybe what microbiome and microbiota have been used interchangeably previously. Even for me previously, before I enter this microbiome projects, I used to use this microbiota, microflora terms. But actually, when we there are some revise in glossary in microbiome journals. The microbiome actually, as what Dr. Hafiz said just now, previously, the entire habitat that includes the microorganisms. Okay, the microorganisms can be bacteria, archaea, lower and higher eukaryotes and viruses. Their genomes and also the surrounding environments. Okay, meaning that microbiome is actually the combinations of biotic and abiotic factors of the given environment. If you are at if the microbiota is in the human, it become the human microbiome. When we talk about the genomes and all that. Okay, this is the differences between microbiome. Maybe I can go a bit. Yeah, the definitions, the entire habitat of microorganisms, bacteria, archaea, lower and higher eukaryotes and viruses, their genomes and the surrounding, that is for microbiome. But for microbiota is the assemblage of microorganisms presented in a defined environment. And for the microbiome, it describes both biotic and abiotic, okay? But for the microbiota, it's only described about biotic factors, where the classification, the taxonomy and all that. Okay, and the microbiome works is mainly focused on genetic makeup of the microorganisms. Okay, while for microbiota, you need to differentiate or identify the type of the microbes. Yeah, and for the experimental methods, microbiome may involve with the functional metagenomics, and we can incorporate it to the multi-omics experiments, for example, metatranscriptomic, metaproteomic, and also metabolomics. While for the microbiota, it's only focused on metataxonomic, which usually use the 16S RNA or 18S RNA amplicon metagenomics. This is the difference between the microbiome and microbiota. Okay, if you can see here, okay, this illustration targeted amplicons, you can see the difference. Okay, the top one is the 16S, 18S, okay, ITS. This is using the amplicon sequencing. The same thing, they use the next gen sequencing, but the target is a single target. While for the shotgun metagenomics, or what we call the functional metagenomics, okay, we use the whole genomic DNA, okay, and target all microbes in that samples. Yeah, and this require a lot of computational analysis, yeah, compared to the normal metagenomics or the amplicon metagenomics. Okay, and there's a if you can see in general, you can see the what is the target actually. If you want to have the microbial potentiality, you need to run the metagenome, metatranscriptome, and metaproteome. If you want to know the microbial functionality, you need to run the metatranscriptome, metaproteome, and if you want to have the microbial activity, actually you need to run the metabolome. Okay, when we talk about the meta here, it's environmental samples. It depends on what type of samples. It can be human, it can be plant, it can be animal, it can be from the soil and all that. Yeah. Okay, that is a bit about microbiome. And as my part is talking about the polar regions, okay, we need to know what is the polar actually. Everybody know about Arctic and Antarctic, yeah, but we go a bit deeper. What is the difference between Arctic and Antarctic? Sometimes people like confused, okay, when they talk about when I said I went to Antarctic, oh, what about the polar bear or something like that? Okay, so this is different between Antarctic and Arctic. Okay, Antarctic is a continental surrounded by oceans. It's a continental, it's a land, yeah, with a volcano system, lakes, and everything. But for Arctic, is ocean surrounded by continents. It's actually ocean, frozen oceans, yeah. And in terms of the annual temperature, the average temperature for Antarctic is like 58, minus 60 degrees. Okay, for Arctic, it can 0 degrees to minus 20 degrees, something like that in average. Depends on the season. You can have the winter seasons or summer season. In terms of the plant and animals, you can see that there's no plant and mammals, okay, terrestrial mammals in Antarctic, but you can find the plants and animals in Arctic, okay. And the most confusion is about a polar bear, as I mentioned, polar bear and penguin, okay. The polar bear is the king of the Arctic, okay, but in Antarctic, the penguin is the king there, right? And in terms of the people and populations, there's no group of native people in Antarctic, but you can find the native people and population in Arctic. Okay, and you can see that the populations of Antarctic is sparse, okay, within the 16 degrees of south, and occurred at the scattered scientific stations, only at the research station or the military stations. But in Arctic, you can find the modern settlements over there. So this is different between the Arctic and Antarctic. Okay, when we talk about the polar microbiomes, okay, this is the major group that we need to consider. Okay, one is the psychrophiles, another one is the psychotrope or psychotolerance. Okay, what is the difference between these two? Okay, a psychrophile is an extremophilic bacteria or archaea which are cold-loving bacteria. They like the cold environment. Okay, optimal temperature for growth is about 15 degrees Celsius or lower, but they can go up to 20 degree maximal. Okay, minimum temperature for growth is 0 degree or lower. But for psychotrope or psychotolerant, they are cold tolerant bacteria, meaning that not necessarily they like it, but they can tolerate. Okay, they have the ability to grow at low temperature. Okay, optimal and maximal growth temperature is above 15 to 20 degrees Celsius. Yeah. So this is the polar microbiomes that usually we study. Okay, in terms of the opportunity, okay, this is the general opportunities. Why you can see, I just want to try to open your mind. Okay, what is the opportunity that you can see, especially for the microbiomes research? Okay, for example, here the opportunity. Okay, I will focus on Antarctic. Okay, because Arctic we have dedicated or respected countries that govern the research over there. So you must have something collaborations with them or permission from them. But the Antarctica is a bit different. Yeah. So, and most of the research for polar research has been done in our Arctic area. Okay. Only not many from the Antarctic regions. Okay, for the Antarctic, okay, it is ice-covered continent, about 98% covered by ice, and some of the places can go up to 4 kilometer thick of ice. Okay, if you can see here the subglacial aquatic system, this one from National Science Foundation report. Okay, you can see that you can see the complex system of rivers and lakes at the Antarctic continents, but it covered by this thick ice. Okay, about 2,000 meters or 2 kilometers. Okay, somewhere. Okay, in these regions. Okay, if it's hidden like this, you know that the potential is high. Okay, you can find the novel microbes over there. Yeah, you can find a novel bioactive compounds over there. Okay, so this is the why we want to study at the polar regions. Okay, and there's another one, a frontier for exploration and discovery. For example, from the NSF report, okay, the surface of Mars is known better than topology beneath the Antarctic ice sheet and its floating ice shelf. For example, here the ocean cavities, which is very important, okay, because that is the meeting regions between the cold, solid ice and also the warm water. Okay, the ocean water, which is warmer than the solid ice. So it's actually completely unmapped and unmeasured. Okay, if you imagine, if you do the microbiomes research over there, okay, it's really have a potential to be explored. Okay, not only that, okay, the Antarctic is a novel records of past and changes. Yeah, so for example, here the sediments under Southern Ocean and Antarctic ice sheet preserve unique records of how the climate and ice sheets have changed in the past. Okay, just now one of the question given to Dr. Hafiz is, can be the microbiomes predictor or indicator. Okay, in terms of this climate change, okay, it can be indicator, because when we talk about the microbiome profiles, we can see different conditions of environment will have a different microbiomes. So it can be like a historic record, or the so from the records then we can predict actually. Yeah, but you can see that here they use the blue ice drills, but this one is only like few meters. It can go up to hundreds meters, few hundred meters if they want with the big machines. Okay, so that all the records of the historic records of the past can be found there in terms of the living organism and all that. Okay, another one is the ecosystem at the edge, where the Antarctic circumpolar current actually form a barrier so that the organisms at the Antarctic regions cannot escape from that area. Okay, so this so that if you want to study the something spatial, then you can go to the Antarctic and find the something which it's only at the Antarctic region, because it's different from Arctic, because Arctic you can go by land or something like that, but this one, because of the system of current, so it's quite difficult for the organism to escape. Okay, so this is the important areas for studies if you want to further the research, okay, in polar regions. Okay, you can go here. For example, how will Antarctica contribute to the changes in global sea level? Okay, what is the role of Antarctica and the southern oceans in the global climate change? And I think for us, okay, even though we can also see the effects of microbiomes in terms of the changes in global sea level, okay, the global climate change, but how the Antarctic microbiome actually responds to this ecosystem to change, yeah, and what is the role as has Antarctica played in changing the planet in the past. So that is some of the important areas if you want to further with the polar research. Okay, for the microbiome opportunities, okay, of course, okay, you can study microbiomes. For example, the human microbiomes, as Dr. Hafiz mentioned. Okay, this every parts of the humans. Okay, they have special microbiomes or microbial communities. Which, when you talk about the microbiomes, the changes in terms of the abiotic factors. Okay, for example, the military forces there. Okay, usually they will come to the station for one whole year. Okay, we can actually compare their microbiomes, for example, in the gut microbiomes, before they go to Antarctic and after they go to Antarctic, because it's extremely cold. So there must be some changes in terms of that, and the gut microbiota need to actually adapt to these physical changes. Okay, same goes to the animal microbiomes. And as what Dr. Zaima said, bioprospecting. Okay, this is very famous nowadays because we try to harness the natural resources. Okay, so if you have the studies on microbiomes of the polar, you can actually use for agriculture. Okay, bioremediations, where my area is, okay, cosmetic and personal care, nanotechnology and biosensors, and also pharmaceuticals. You can find the antimicrobes, okay, antibacterial over there a lot. Okay, there are several articles which actually manage to isolate antimicrobial compounds, okay, from the polar microbes. Okay, another one, maybe the climate change and environmental protections. We know that the microbes involved in biogeochemical cycles. Okay, the microbes, the microbiomes of the polar, which is a very big area, there must be some contribution to the biogeochemical contributions or cycles. Okay, and also the global warming. Okay, so this is some of the examples, the cold active or adaptive enzyme in the industries, the applications, for example, here in the dairy, okay, of F&B, fruit juice, okay, meat, okay, this is what I'm trying to do now. We have the cold active protease isolated from Ross Islands. So we try to use the meat tenderizer. Okay, what is interesting here for the meat industries? Okay, for example, if the working people, okay, usually when they come back from work, they want to cook meat, they need to thaw it, okay, or put the tenderizers outside. Okay, but here, if you use the cold active protease, you can actually tenderize the meat while storing in your fridge. Okay, at a cold temperature, it's still active. Yeah, so when you come back from home, you just take it and you straight away cook. Okay, easy for you. Okay, for examples, again here. Okay, the textile. There are also cold active proteases to improve the productions. Yeah, and also fabric finishing. Sorry. Okay, and also you have this cold adapted cellulase for biopolishing and stone washing processes. And as for us, molecular biologists, okay, this is some kind of there are some irreversible enzymes in activations by mild heat treatment. You can actually inactivate the enzymes that are used, not at a high temperature, okay, at a lower temperature, which will actually will not interfere with the subsequent reactions. Okay. And for biomedical, for those who involved in biomedical, there are a lot of biomedical, especially bioactive compounds, something like that, isolated from the polar regions. Okay, for one of the examples is alpha galactosidase, which remove the antigenic components from surface carbohydrate. Yeah, and also you can have this the cold active alpha galactosidase from pseudoaltromonas. Yeah, and also the cold active nitro reductase. Okay, this is another part of my projects, okay, which involve the bioremediations. I use the sacrophilic microorganisms, okay, because one of my projects in Antarctica is actually to treat the diesel polluted soils. So we try to study the taxonomic or the microorganism or the community of the microorganism, and also the genes that involve in the degradations of this polluted of the diesels. Yeah, and if we can have that, actually we are very it's a very good because we can use this enzymes or microbes, okay, to treat the polluted soils, or especially the diesel, as I mentioned, okay, at the winter or the cold environment, okay, especially if you have this at the open oceans, which is a colder area. So it's very difficult if there is some oil pollution, something like that, to be treated, because the coldness of the area will reduce the activity of the microbes. Okay, but if you have this sacrophilic microorganism or cycrotolerant microorganism, you can use it. Yeah, to and also the bioaugmentation and inclusion of contaminated environments. Yeah, to help in biodegradations of the recalcitrant chemicals. There's some applications of the bioremediations. Okay, in terms of that is the opportunities. Okay, you can think now what you want to do, but there is some challenges of and the challenges I can divide into two, especially for the microbiomes. One is the polar research itself. Okay, the most challenges is the accessibility to the study area. Okay, if you go to Arctic now, I think it's very easy because it's through respective countries within the Arctic circles. There are about 8 of the countries, okay, in the Arctic circle. So if you want to go to the Arctic, you can go through these countries. But for the Antarctic, because there are not connected, okay, to the main populations of people in the world. So only several countries which a member of Antarctic Treaty, we can go there. Okay, if you can see here, Antarctic Treaty consists of 54 states party, where 12 with consultative status, meaning that they have a voice to vote, and the 7 of it claims the portion of Antarctica as territory. For example, here, if you can see the parties with consulting status making a claim, you can see that Argentina, okay, the Chile, Australia, and also New Zealand. Okay, so that is the consulting status making a claim to Antarctic territory. Okay, and there are also parties with consulting status reserving the right to make a territorial claim. Okay, in yellow color, you can see that US, okay, and also Russia. Other parties with consulting status, the blue color, okay, like, okay, in Asia, we have country like Japan, Korea, China, and also India. Okay, this is the countries that have stations in Antarctica. Okay, parties without consulting status, the green color, like Malaysia. Malaysia involved with this treaty, but without consulting status, meaning that they cannot vote. Any decision about the polar research or the treaty, Malaysia cannot vote. Okay, and the non-party UN member states and observers, the red color, they are observers because in the treaty they want someone who outside the parties, okay, which can be observer. Okay, so that is the accessibility. So you need to have the collaboration with them, okay, for you to go there and use their stations, all that. The second challenges is the cost. Of course, the traveling is very expensive. Okay, for example, this is what I give here is the commercial transportations. Yeah, for traveling for 6 to 10 days voyage it will cost average about 10,000 USD. Okay, and most economic is only about 5,000 USD. Okay, but for the researchers, usually we will have collaborations with the countries that have the stations. Once they have the station, usually they will have their transportations. Okay, for example, in my case, I work with Chilean University and Chilean Ministry of Defense. So we have the access to their Navy ships and also the Air Force, okay, to bring us to Antarctica. Okay, and in terms of the accommodations at the research stations also is one of the costs that you need to consider when you want to do the research over there. Okay, if we don't have the proper collaborations with the country that have stations over there, you can pay actually the accommodations. Yeah, so but it's not cheap, right? And the other cost is meals. Okay, you cannot cook, you cannot bring the raw materials. Okay, so they will cook for you, they will provide food for you. The problem with a Muslim, okay, because most of the countries and the stations there is non-Muslim countries. Okay, so we have problems with the halal food, so we need to bring the stocks of halal food for us. Okay, and yes, sometimes they give us fish, okay, like salmons. I eat salmon every day. Okay, so this in terms of the meals. Yeah, the another challenges is limited communications. Okay, some stations will have a very good internet networking. For examples here, okay, this is Kim Sejong Korean stations. Okay, so they have very good internet networking. But in my case, I stay at an isolated island with a small stations. We have very limited internet access. So if you want to open one email, actually it will takes like hours. Yeah, so it's not totally no communication, but it's very limited. Okay, and of course, if you go to the polar regions, it is extreme environment. You need to adapt with the cold environments, of course. Okay, in terms of the challenges in microbiome studies, I can give like two parts. Okay, experimental challenges and also computational challenges. Okay, in terms of experimental challenges, for example, the study designs, okay, the sample size, the controls, what you need to do, the cohort selections, in terms of the sample handling, transportations, storage, is very difficult for you, especially for the polar microbiome. Okay, I will talk about later about the transport and storage. Okay, we need to get the permissions to bring the samples and all that. Okay, another experimental challenges is the nucleic acid preparations, the extraction method that you use. Okay, barcoding and adapter ligation, amplification, library preparations, because this is involved with the cloning. Okay, and also sequencing. There's a lot of machines over there, the NGS system. Okay, which one you want to choose? Okay, how you want to run the quality assessment? Okay, sequencing that the coverage of the sequence that you want, and of course the cost, because it's not cheap. Yeah, once you have these environmental challenges settled, then you have the metadata. Okay, once you have the data, you need to analyze using the computational approach. So the computational challenges you need in terms of the quality control. For example, here, if you have the low quality reads of the sequence, what you need to do, okay, and how you want to separate from the host sequences, because you're during the library preparations. Okay, so once you have all the sequence, which one is the host sequence, which one is your real sequence, your genome sequence that you want to have. Okay, and also the contaminating sequences, and which how you want to trim the sequence and all that. Okay, the next one is the assembly. Okay, you can have the repeat regions. Okay, so and also the sequencing errors. These all the problems in the computational analysis. Okay, and how you want to choose the good algorithm, because there is a lot of algorithms for the assembly. Okay, and then the taxonomy. Okay, the database. When you talk about the microbiome, you can go from the meta taxonomy to the functionals. Okay, metagenomics. So the taxonomy is one of it. How you want to refer to the different databases. Okay, how you want to predict the genes, the alignment, the clustering, and also the binning. Okay, and the last one is the functional classifications. Okay, so which algorithm for you to use for annotations and all that. This is some computational challenges. Okay, this one I already covered just now, the experimental condition, computational intensive. Okay, lack of standardization in laboratory computation, and the most important is biosafety issues, because this will depends on the policy, law, and regulations of the countries. Okay, if you want to bring your samples for Malaysia, if you want to conduct the polar research, you need to get permissions, okay, from Department of Agriculture to bring the sample, especially the soil sample. Okay, the import permit, everything is need the permissions from the authority. And most important things, okay, the unknown microbiota need to be considered as high risk. Okay, it is unknown. Who knows that the samples that we will bring something that highly pathogenic, okay, like the COVID-19, who knows? So we need to regard it as a highly risk samples. Okay, so this is biosafety issues. Okay, with that, I think, thank you. Salam Ma'al Hijrah and happy Merdeka Day. Dr. Akbar, it's up to you.
Thank you very much, Dr. Faisal, and it's one of the interesting presentation, and you brought us back to polar region. And if there is any question, the participants can ask. You can just text so that I can unmute you. Maybe I'll start my question, Doctor.
Okay, sure.
Regarding the psychophilic microbes in the Arctic regions or Antarctic region, as you mentioned in your presentation, that it can grow better in the temperature between 15 to 20 or within this time. But when we use it for the bioremediation process of oil spills or whatever related, what we can feel is in the tropical region, the temperature of the sea is at least 25 to 26. So, how possibly we can use the psychrophilic bacteria, which are isolated from the Antarctic region, polar region, to be used in the area of tropic?
Okay, thank you for the question, Dr. Akbar. As I mentioned, there are two group of microbes, the psychrophilics and psychrotolerant. Okay, which the psychrotolerant usually, if we want to use to the higher temperature, we can use it because it's the optimal temperature is 15 to 20 or 25, but it can go further up. So there are some special cases where we actually can grow the psychrotolerance up to 40. Okay, so because they actually, it's not the psychrophilics, they are psychrotolerant. They can adapt to the lower temperature, something like that. Yeah.
Oh, fine. The question number 2 that I would like to ask, if there is no question from the audience. So the second question is again the psychrophilic or psychrotolerant bacteria which are isolated from uh, polar region. When they are usually when we want to use the bioremediation processes, usually we isolate the samples from the place where oil spills are there or where the pollution is predominant.
Yeah.
So in terms of Antarctic or Arctic region, we do not have any sort of pollutions there. So how possibly can we use that particular one in this area?
Okay. Actually, when we talk about the pollutions, Dr. Akbar, if you familiar with the Arctic regions, there are a lot of oil explorations over there in the Arctic. Yeah, and there's a lot of potentials of spill. So that is why we want to use the psychrophilic or psychrotolerant to do the bioremediation processes. Okay. And for the Antarctic, actually, there are some spill cases. Okay, for example, the diesels. Okay, I cannot say which stations. Okay, because this one is a confidential. Okay, so but there are some diesel leaking from the tank to the seep through the soil and through the marine environment. So this is what we actually trying to solve.
Clear, doctor. Thank you very much, doctor. And I'm waiting for the question from the audience if there is any. And if there is no question, we respect the time of the presenters as well as the audience. We plan to finish it by 4 o'clock. Now 4 o'clock, 3 minutes already. So if there is no question, we shall wind up our webinar. Thanks to all the presenters for your intellectual presentation, as well as all the participants from Zoom platform, as well as the YouTube live streaming. Regarding the presentation slides, I will humbly request all the presenters to share their links to me if they're willing to do so. And these presentations will be shared in IIEM Marine School webpage. So those who wants to join, to have an access to it, make use of it. Thank you very much. Without further, since there is no question, I would like to end up the webinar session with the tasbih kafara. Subhanallah bihamdihi subhanallahumma bihamdika ashhadu an la ilaha illa anta astaghfiruka wa atubu. Thank you, and we welcome you to join the next webinar, which we will post in the IAM community. Thank you very much.
Okay, thank you, Dr. Abah. Assalamualaikum.
Dr. Faisal, thank you. Thank you to Dr. Zaima, Dr. Hafiz.